So the first part of the study guide, which you can see here, okay, is the list of definitions. I'm not going to ask you definitions directly. Okay, you don't have to write the definitions in the exam. It's all multiple choice, as I mentioned before. But I don't think it's, I think it's useful just describe each of these terms in a short phrase. Okay? Like the example is Buffy code, the part of the formed elements that contains platelets and white blood cells. We move on. Okay? We have all those definitions in the lectures. And obviously, you know, like you can go on and on about many things that are listed here, but you have to figure the most important, the ones that I've highlighted basically. Like I tell you, platelets, what do? They form clot, they regulate inflammation. You're moving on. Okay? You will have a chance to come back to them in another part of the study guide. Am I clear? So I want to clear out some things that we never mentioned. We never mentioned defensins. So it's not going to be on the exam, obviously. Mention respiratory burst. Mention this. This three proteins, ferritin, hemosiderin, transferrin, don't. Okay? Just don't. Not going to ask about those three. Okay? And don't worry, if you don't catch it now, it's all recorded. It's all going to be on YouTube. Okay, so ferritin, hemosiderin, transferrin, don't focus too much on that. Extravasation, intravasation, adhesion molecule, diapedesis, cytokine and chemokine. I want so it doesn't puzzle you like how deep should I know that? Cytokines and chemokines are the molecules that direct white blood cells to the site of inflammation. That's as far as it goes. Am I clear? Hemopoiesis, you'll get this words. Yeah. Red blood cell production. Hematopoietic stem cell. All you need to know about this one, that's the cell from which all other blood cells originate. Are we clear? Colony stimulating factor. Screw it. Leukemia. Abnormal proliferation of white blood cells. That's as far as it goes. I'm just checking what we... Oh, crap. Sorry about this. Of course, intrinsic pathway and common pathway. Does that make sense? So you have to know, intrinsic, we have extrinsic, they all come together to the common pathway. Okay, we discussed tissue factor, prothrombin activator. One very important thing, I want to say it now before I forget. Remembering some conversations before. Remember how I told you to picture the pathway? Like you start the exam and you want to put something on the paper and you do it, and perfectly fine with me. Okay, it's okay. Do not come to me during the exam showing me the pathway and asking me, is that correct? I made this mistake once. I'm not going to make it again. Here's the deal. It may not be entirely correct, and in this case, I will be obliged. I mean, I would say I have no idea. you're on your own, okay? However, like if you come to me and say, is it correct that prothrombin activator becomes prothrombin? I kind of know what you're talking about. I know that you're referring to the fact that prothrombin activator converts prothrombin into thrombin. But if you will come and ask me, does it become prothrombin? I will be obliged to say no, it doesn't. And I know what you're trying to figure out. I know that you have the pathway kind of in the correct order. I don't want to cause this confusion, so please be careful with terms like, does it become something? Because I take it very seriously, okay? If you tell me like, Papa, the daddy bird and mommy bird, they become a baby bird? No, they don't. They do not become the baby bird, okay? So if you ask me a question, I will try to keep away from this as far as I can. I'd rather say I cannot answer. They will direct you in the wrong way. Okay, deal? As I vividly remember this whole ordeal about it. Aspirin, warfarin, the big trend. All you need to know about those guys is that they are anticoagulants. Am I making sense? I'm not asking like, would you prescribe a person with stage 1 peptic ulcer who is at very high risk for the stroke? Would you prescribe this person an aspirin? What the fuck do you know? No, for sure. So a plasmin. So tissue plasminogen activator activates plasminogen. Plasminogen is the result of this activation. Plasminogen becomes plasmin. Plasmin destroys the fibrin threads. Again, antithrombin. Well, kind of name explains it all, right? Know the roles of nitric oxide prostacyclin they produce by healthy endothelium. What petechiae is disseminated intravascular coagulation. Know what happens during these things. Does that make sense here? Okay. I'm not going to ask you about what is the most common cause of disseminated intravascular coagulation. No. If I say this, you tell me that's when the blood clots in the peripheral blood vessels, disseminated intravascular coagulation, which immediately makes the blood more leaky because all clotting factors are removed, which leads to the massive hemorrhages with the massive thrombosis. Kind of two things that usually do not go together. Hemophilia, agglutinin, agglutinogen. So agglutinins, remember agglutinins are antibodies like anti-B, anti-A. And agglutinogens are A or B. The agglutinogens are present on the surface of red blood cells. Rhesus factor, just know that it's one of the blood antigens. That's as far as it goes. Hemolysis, breakdown of red blood cells. Rhesus conflict. For rhesus conflict, you saw the question today? You saw the question today about rhesus. That's, know the mechanism. And erythroblastosis fetalis is the condition that can stem from the rhesus conflict. That's it. Protective chemicals. Again, if I ask you which of the following is, know where they found, okay? It's most important, where they found. And these two are, antibacterial proteins. By the way, this was my first experiment with an extended study guide. So then I had to curb my appetite a little bit in terms of the pages. Yeah, again, defensins, screw defensins. For white blood cells, so we get in there. It's probably going to be repetitive, And if I explain something now, I hope you don't mind that I'm going to be skipping it next time. We good? Clear? Okay. So for white blood cells, what you need to know, what they are active against and what they can do. So like questions that you saw today, if I ask you neutrophil, you tell me, oh, it's bacteria. Makes sense, right? Two main phagocytic cells are macrophages and neutrophils, remember that. When I say what they can do, I refer to mechanisms, like neutrophil can produce respiratory burst. It can release neutrophil extracellular traps, and it can phagocytose. Eosinophils, can they phagocytose? No. Basophils, can they phagocytose? No. Mast cells, same. No. Sorry, agranulocytes. To know which ones are granulocytes and agranulocytes. So if I ask you, what is the granulocyte that can phagocytose? You tell me that's neutrophil. You clear? For NK cell, often, it's kind of mixed up. Remember, NK cell, can it phagocytose? No. What does it do? It kills cells by inducing apoptosis. Okay? So NK cell is somewhat similar to cytotoxic lymphocytes. Okay? The difference is cytotoxic lymphocytes are highly specific. That makes sense here. Cytotoxic I explained this. Cytotoxic lymphocytes are highly specific. NK cells will be able to tell apart cell that is generally infected versus cell that is not infected. So they will try to destroy all cells that appear infected, no matter what they are infected with. Cytokines, chemokines, again. So yeah, inflammation. For the inflammation, you need to understand that mechanism that we discussed. Inflammatory cytokines, chemokines enter the bloodstream, act upon the bone marrow, induce leukocytosis, right? Elevated number of leukocytes. Those leukocytes, mostly neutrophils and monocytes, enter the circulation. Then they recognize the site of inflammation by adhesion molecules. They roll, they adhere, they go through diapedesis and enter the tissue, and then following the positive chemotaxis, they get to the site of inflammation. Very cool. I think we're pretty cool. Vascular permeability. I'm not going to ask this. You all understand what it is. Okay, this huge thing, actually, I'm cutting you a lot of slack here because I love this topic. We didn't talk about it. So none. We talked about phagocytosis. Basic steps. Pseudopodia formed. Phagocytose the pathogen. Bring it in the phagosome. Phagosome fuses with a lysosome. Lysosome delivers the destructive enzymes and chemicals. And I have a brain freeze, I'm sorry. And microorganism dies. And then the debris is released via exocytosis. Are we good? Okay, I'm just checking on the back row. Okay. For complement, three pathways. I'm sorry. So it stimulates each other. So classical pathway is going to be Antibodies, lectin pathway, remember it's a sugar called mannose. And for alternative pathways, some chemicals on the bacterial surface. Opsonization, when something covers the surface of a pathogen, making it more attractive to macrophages. Does it make sense? Do you get it? Okay. So, complement can opsonize. It can form membrane attack complex, and it can also increase inflammatory responses, can stimulate inflammatory responses. Interferon produced by infected cells, protein, warns other cells about incoming infection. Clear? Receptor, I kind of just know that the interaction with the cell happens through the receptor. I never specifically mentioned that, but we spoke about it on so many occasions. Okay. Difference between innate and adaptive immunity. So innate immunity, given adaptive immunity, requires exposure, cellular, be it T cells, humoral B cells that produce antibodies. Know what bone marrow and thymus are. T cells, B cells, two types of T cells, the function of T regulatory. I'm going through because this is a straightforward explanation. So plasma cell produces antibodies derived from B cell. Okay, T killer cell, cytotoxic lymphocyte kills infected cells derived from CD8 T cells. Does that make sense? You do things like that. Good? Yeah. This, so complete antigen, haptene, antigenic determinant. Sorry. Complete antigen. Protein, DNA, complex carbohydrate. Haptene, small molecule that you can attach to the carrier. Are we clear? And then it's going to produce immune response. Antigenic determinant, a fragment of a molecule which is recognized by the specific antibody. We didn't talk about positive selection. Never mind. Negative selection, when cells that can recognize our own antigens are destroyed. Remember that conversation we had? Self-reactive cells are destroyed. Now for B cell receptor, know that it's like an antibody, but it is attached to the B cell, and it recognizes the antigen. Structure of the antibody, to the level of 2 heavy to light chains, 2 FAB fragments that bind antigen, and 1 FC fragment that binds to the cell. So antigen binding side, the FAB, right? VDJ recombination. Remember we talked about the mechanism how antibodies can can recognize so many antigens. That's what I refer to. You have to understand how it happens. By how it happens, I mean all you need to know, we have different genetic fragments, and they recombine into the unique sequence okay, and produce unique, pretty much unique FAB part of the antibody. Does that make sense? When you combine like certain V, certain D, certain J. 5 antibodies here. Know the function. I'm going to ask you about this. Primary and secondary immune response. Secondary immune response is more robust, happens faster, because it involves memory cells that were formed during primary immune response. Okay? Primary immune response is when you first time is exposed to the pathogen. Active versus passive, artificial versus natural. You saw the question today? Something like that. Okay, I'm going to ask you this. Mechanisms by which antibodies work. So they can neutralize a glutamate precipitate, right? Can they opsonize? Yes, they can opsonize, called opsonization. So if I ask you, what can opsonize? Can antibodies do that? Yes. Can complement proteins do that? Opsonize. Can complement proteins opsonize? Yeah, that's one of the mechanisms. Increased inflammation, opsonization, membrane attack complex. Okay? Can interferon opsonize? Interferon? No. Does that make sense? So you need to know that there are 2 main mechanisms of pathogen opsonization. It's complement and antibodies. Does that make sense? Any questions so far? Don't hesitate to interrupt me. MHC. I want you to know the location. Class 1, all nucleated cells. Class 2, only antigen-presenting cells. Am I clear? I also want you to know so each class, what does it present to? Am I clear? So it presents to MHC class 1, presents the antigen to the CD8 cells. MHC class 2 presents the antigen to the CD4 cells. Okay, so basically CD4 cells, what they can recognize, which cells they can react with, since they recognize only CD4. Say again? No. What I'm saying is, I probably didn't phrase. So, MHC class 2 presents the antigen to CD4 cells. Therefore, CD4 cells will interact during the antigen presentation with antigen-presenting cells, macrophages, or B cells. 3 types of APCs, right? CD8 cells, in general, can interact with any nucleated cell in the human body. Does that make sense? So for antigen-presenting cell, know the 3 types: macrophages, B cells, dendritic cells. Okay, so this, Fas receptor, Fas ligand, perforin, and granzyme. Remember we were talking about the mechanisms how, lymphocytes, cytotoxic lymphocytes, or natural killer cells can destroy the infected cells? So there are 2 options. One option is Fas receptor and Fas ligand. You don't have to memorize where is Fas receptor, where is Fas ligand. But Fas receptor and Fas ligand interact. And that initiates apoptosis of the infected cell. So that's going to be one mechanism. Another mechanism is when perforins perforate the membrane and allow granzymes to enter the infected cell and kill it by inducing apoptosis. So again, coming back, which two types of cells use this mechanism? Perforin and granzymes. One type in adaptive response, one type in innate response. They're both killers. Perforins and granzymes, no phagocytosis involved. I heard something. So T cells, cytotoxic lymphocytes, bless you, that's adaptive. In innate response, there is a cell that uses the same mechanism, just not specific. It's also a killer. NK, natural killer cell. Does that make sense? So they both use the same approach. Vaccines. Basically the definition. Did you see the question today on the vaccine? I'm going to give you a description. Vaccine. Okay. I don't know. Vaccine against influenza is influenza virus that cannot replicate anymore. It's completely inactive. So which one is that? Inactivated vaccine. Well, I'm not going to ask this. You understand, But say if I ask you, okay, the flu mist is the vaccine against influenza that contains influenza virus that's weak, cannot cause infection. Type is that? That's attenuated vaccine. Does that make sense? That's as far as it goes. Hypersensitivities. Mechanism. We can review them now. So in hypersensitivity type 1, what happens? Which antibodies mediate type 1 hypersensitivity? IG. Is it fast? Okay, so it's immediate. Do you get response during your first exposure to the allergen? No. During your first exposure, you form antibodies. IgE. IgE, where do they bind to? Which cells? M-word. When they are formed, mast cells, yes, and they stay bound to mast cells, waiting for the second exposure. When second exposure occurs, an antigen binds to the IgE that are on the mast cells. What happens to mast cells? They release histamine, they degranulate. Does that make sense? Now, type 2, which antibody is mediated? Ig. G, immunoglobulin G, and the prominent example of type 2 is transfusion reactions, right? So in transfusion reactions, they basically just bind to the blood cells. Type 3, it's specific type because in type 3 cells form, antibodies form immune complexes with antigens, and those immune complexes, where do they deposit? Deposit in the basement membranes of epithelial layers. So your immune system attacks that basement membrane and basically, destroys the integrity of epithelial cells. That makes sense. And finally, with type 4, so it's T cell mediated and it is delayed. It takes a longer time to develop because it's T cells. After the first exposure, you produce T cells specific to this allergen. And during the second exposure, those T cells, T helper cells, they recognize that allergen, get activated, activate cytotoxic lymphocytes and macrophages, and you have this horrible inflammatory response in the site where the allergen is present. Okay? Makes sense? Difference between different types of grafts. From where to where? Just, that's it. That's all I'm going to ask. Okay. Graft versus host disease. In the graft versus host disease, what happens? Can you tell me? What responds to what? What type of transplantation is it? Huh? Bone marrow. So what attacks what? Not the body. What specifically attacks the tissues of the... So in relation of donor-recipient, what attacks what? T cells that come from donor bone marrow attack tissues of the recipient. Are we clear? Because it's graft versus host. So graft attacks host. Does that make sense? In the rejection, it refers to what type of transplantation? What type of organ transplantation? Call it solid organ, right? So it's solid organ transplant. And in this case, what attacks what? The recipient attacks the organ from the donor. Does that make sense? Clearly know the difference. Immunodeficiencies. Be able to distinguish congenital and acquired. I practically gave you a link of these 3 names that you need to know for congenital immunodeficiency. I'm not what I can ask. Indeed, George, what you don't have, no T cells, right? In A gamma globulinemia, no B cells. And in SCID, no T and B cells. Okay, we're good? Acquired immunodeficiency, different types of disease. Lymphatic vessels, we'll get back to it. Okay, what I think we should do, I'm going to just scroll all the way down, just to clarify. Okay, so we're going to reiterate it a little bit just to make sure everything is clear. When I say blood composition, you should be able to list the components of the blood. So, plasma, from the elements, including red blood cells, white blood cells, platelets. Okay, I do expect you to know that. Okay. Major components of the plasma and their functions. The terms that I showed before, alpha, so albumin, water, alpha, beta, gamma globins. What's the function of alpha, beta? Transport. Gamma, antibodies, yes. I mean, a lot of things are really basic. If I ask you, is there any oxygen in the blood? You're allowed to tell me duh. Okay. So I'm kind of focusing on more crucial components. Okay. When I say structure and function of erythrocyte and hemoglobin, erythrocyte has biconcave shape. Hemoglobin has 4 components in it, right? 2 alpha to beta chains. How many heme molecules are in one hemoglobin? 4. How many irons? 4. One per heme. Yes. I see where you coming from. Yes. 1 per heme. So how many oxygens? Maximum. 4. Is there any? It's kind of embarrassing to address my own mistakes in the quiz, but is there any hemoglobin in the plasma? Normally. No. No. There's no hemoglobin in plasma, it's entirely in the red blood cells. Okay? I know. Oxyhemoglobin, where is it formed? Lungs. Deoxyhemoglobin is formed in the tissue. So oxyhemoglobin has oxygen, deoxyhemoglobin doesn't. Erythropoiesis. So for erythropoiesis, what you should focus on. Which hormone stimulates erythropoiesis? Erythropoietin, which is produced by kidneys. Erythropoietin is produced by kidneys. What can stimulate erythropoietin production? Hypoxia. What else? Hypoxia directly. Anemia, blood loss, any instance in which you have less number of functional red blood cells. That makes sense? When I say how body maintains the iron levels necessary for erythropoiesis, screw that. I mean, we eat it, okay? Just that's it. We eat it. Stored and transported. I told you I'm not going to ask about hemosiderin, transferrin, and ferritin, so you don't have to focus on this. Okay? Vitamins necessary for erythropoiesis. Let's just cut it out. I mean, sounds it's a lot. It's a lot. So I try to remove little tiny tidbits, but basically you need folic acid and in B12. In terms of clinical practice, when you get to actual patients, remember, red blood cells come from rapidly dividing cells. If folic acid is necessary for nucleotide synthesis and B12, I don't remember for what part of the cell metabolism, but we need it for cell division. Without these two vitamins, bone marrow cells cannot divide. Since they cannot divide, you're not going to have any new red blood cells. Did I make myself clear? Okay. After RBCs expired, they go where? Spleen. Die there. Sickle cell anemia, thalassemia. Mutations, associated health problems, many. Focus mostly on sickle cell, okay? Because we discussed it more. I can ask you what happened. Like, which of the following happens in a sickle cell anemia? I don't know. Patient's brain melts. No, it doesn't. But red blood cells get damaged. If red blood cells, can patient with sickle cell anemia develop hypoxia of tissues? Yes. Hemolysis? Yes. Damage of endothelium? Yes. Evolutionary benefits. I refer to malaria. So far, am I making clear points? Two major types. That's granulocytes, agranulocytes, how they migrate into tissues. Describe the process step by step. That's inflammation that I referred to. Leukocytosis, rolling, adhesion, diabetes, positive chemotaxis. Good? Explain leukopoiesis. Well, there's not much to explain, or you can write like a 2-volume textbook on leukopoiesis. What I'm asking is, you have inflammation, you have inflammatory mediators, they stimulate leukopoiesis. Does it make sense? So white blood cells are produced in response to, say, infection. Cells of myeloid versus lymphoid origin. Cells of myeloid origin, where do they end up? In the blood. Cells of lymphoid origin end up in lymphatic. Good? Structural functional differences. We talked about it. Again, I would focus not so much on bilobe nucleus or trilobe nucleus or whatever lobe nucleus. I would focus on the function. Eosinophils, type parasitic, and so on and so forth. Main pathologies associated with white blood cells as far as leukemia goes. That's it. Generation of platelets. Remember thrombopoietin? Remember that absolutely fascinating story that thrombopoietin binds to platelets? Remember, no? So we're going to call it TP, if you don't mind. So you have thrombopoietin plus platelets, and they form complex. Okay? So if you have a lot of platelets, do you have a lot of thrombopoietin in the plasma? No. If platelets are gone for some reason, what happens to thrombopoietin? Thrombopoietin in the plasma goes up, stimulates platelet production. A lot of platelets, thrombopoietin is bound. You have low thrombopoietin in the plasma again. Does that make sense? Know this mechanism. Hemostasis. What do we start with? Yes. So we damage the blood vessel. There's blood loss. What happens to the blood vessel after? Immediately after. It constricts, right? Now, when you have a blood vessel damage, there's an exposure of collagen. What happens to platelets? They stick to it. They get activated because they interact with von Willebrand factor, okay? And they stick to the vessel wall, damaged vessel wall. What happens to the platelets that are frivolously swimming through the blood flow, through the blood, that are not stuck yet when they reach the site of the damage? They get stuck too. They get activated, right? And then they activate other platelets and other platelets and other platelets. So which type of regulation is that? Positive feedback, right? That makes sense? Why platelets do not stick to the endothelium that is not damaged? Nitric oxide and prostacyclin, right? Well, we're not supposed to drive 10 miles over the limit. Okay, we still do that. So they don't stick because there's prostacyclin and nitric oxide that are produced on the endothelium. It's not damaged. Does that make sense? Good. They prevent platelet activation. So you have a platelet plug. Now, why coagulation is so important for fixing the damage? Huh? No, what I'm saying is you already have a plug. We're talking about specifically coagulation. You have a plug. Isn't that enough? What do I want to do with a plug? You have to hold them together, okay, with fibrin threads. So for this we have coagulation, okay. Two pathways, intrinsic and extrinsic. Intrinsic is slower, extrinsic is faster. Intrinsic is stimulated by the factors inside of the blood vessel, right? It can be platelets, it can be collagen, it can be any protein that comes from the damaged endothelium or basement membrane. For extrinsic, what is the stimulating factor? It's called tissue factor. The steps that I want you to know, in intrinsic it's 12, 11, 9, 8. In intrinsic, in extrinsic, sorry, it's basically 7. Okay, 7 plus tissue factor. And they all come down to the common pathway. They converge on the first element of common pathway, which contains the coagulation factor 10. Does that make sense? So, and that's what starts the common pathway. Now, coagulation factor 10 together with some other stuff, factor 5, calcium, spatulocerium, just know that the common pathway starts with the formation of prothrombin activator. Prothrombin activator, what does it do? Activates prothrombin. That is the moment when scientists are really smart in naming. Because I think that the choice of names for coagulation factors exemplifies the poor judgment. Like, factor 12 says nothing to me. Okay? So, prothrombin activator activates prothrombin into thrombin. Thrombin activates fibrinogen into fibrin. Fibrin forms mesh. Correct? Now, let's talk about the regulation and control of the clot formation. So we figured why platelets do not stick to the healthy endothelium, right? What limits the growth? So that limits the growth of platelet plug. Why we do not lose all fibrin from the blood in 1 second? When fibrin forms, what happens to thrombin? It binds to fibrin, remember? So more fibrin, less available thrombin. On top of that, there is an activation of tissue plasminogen. Tissue plasminogen activator activates plasminogen. Plasminogen becomes plasmin and starts to destroy the fibrin threads. That's later. It's a clot retraction. Now, antithrombin and heparin, they control the thrombin activity, right? And prevent the spread of coagulation cascade all over the blood. Very elegant method by flowing blood. When blood flows is basically, washes away and dilutes the clotting factors, not allowing the massive spread. Does that make sense? Anticoagulation drugs and their mechanisms. I'm sorry. If you want to know what's going to be on the exam, who should you ask? Me. So I'm going to give away not the question, but the type of the question. I am not going to ask you what is the mechanism of anticoagulation action of aspirin, colon. And then you have to pick. I'm not going to ask this. I am going to give you a mechanism. Okay? I'm going to tell you. Aspirin inhibits the enzyme that produces thromboxane A. Thromboxane A can plays a role in activation of platelets. Which of the following you will see if you administer aspirin? Okay, you can say lack of platelet activation, so no platelet plug is formed. Or I can ask you, thrombin activation, tell me, no, thrombin is going to be fine because thromboxane A has nothing to do with thrombin. Does that make sense? So you will have to figure out which of these steps is blocked. Am I being clear? If I'm not, I don't know if I am actually. So tell me, please. Do you understand what I'm saying? So I give you a full description of mechanism. And you have to figure out which event is not going to happen of the listed. Do I make sense? Do you understand? Like, it's like, let me use another example. It would be a question like, imagine you get rid of all neutrophils. What type of pathogens you will be very sensitive to? You get rid of all the neutrophils. Bacteria. Does that make sense? Because you have no protection against bacteria anymore. Does that make sense? That's the type of thinking that you will have to employ there. Intrinsic and extrinsic pathway, do they end up with the same stuff? Yes. Hemophilia, thrombocyte, again, hemophilia. Remember that hemophilia is the lack of clotting factors. That's it. I'm not asking about which one is which. Thrombocytopenia, low platelets. Blood types. We'll get a question on the blood type. Something like what I asked you today. Does that make sense? So I could ask you something about, what can you transfer to the patient like this? What can you transfer to the patient like this? That's as far as it goes. Am I clear? Okay. Know that, again, agglutinins are antibodies, agglutinogens are antigens on the red blood cells. Explain the rhesus factor. What I'm referring to is, of course, know the mechanism of rhesus conflict. If I am rhesus negative, do I have antibodies against rhesus factor? I presume that I never received any transfusion ever in my life. I do not have antibodies. They are not preformed. Does that make sense? Remember that. But if I receive a transfusion from rhesus positive person, will I make antibodies? Yes. Good. We're clear about it. This top one, chemical mechanical barriers, are they more or less clear? Which ones are present where? I hope I was pretty clear. Cilia, this specific question I want to address. When you smoke, cilia is destroyed. What happens then? What is impaired? Movement of what? Mucus. As far as it goes. Again, blood cells, innate adaptive, so inflammation, it kind of reiterates, yeah, which cells? Two cells that participate in the general inflammation the most. Basically, phagocytes. Which two phagocytic cells we have? Neutrophils, macrophages. By the time we're done with the review, you're going to wake up during the night with a nightmare, screaming neutrophils and macrophages. Phagocytosis. Do you understand the difference between vesicles? Okay. Phagosome is just sort of encapsulation for the microbe. Lysosome is the one that contains actual destructive chemicals. Be good. Oh, how macrophages can tell pathogens from non-pathogens. I said it wasn't too long of a statement or description, but chemicals on the surface, chemical signatures on the surface. Okay. We didn't talk about how pathogens can pathogens survive phagocytosis and how pathogens can exploit that. So just know the difference. Actually, just sort of a note on the side. There's a great microorganism that is called Klebsiella burnetii. It's a tiny little bacteria. It survives not just in a macrophage. It lives and reproduces inside of the phagolysosome. So it gets eaten. Imagine like you being eaten by a giant shark, and forgive me for unbelievable circumstances, but you start to reproduce inside of the shark's stomach successfully. Kind of hard. That's what this bug can do. Pretty awesome. Complement pathways. What's common between them? What's common between them? They all come to the C3b convertase, and then they produce three, I count three major mechanisms: opsonization, increased inflammatory responses, and formation of membrane attack complex. Okay, clear? Good. Interference, major types, that's easiest, because major types, type 1 and type 2. Two types. What triggers their production? Infection. What do they interact with on the cell? Receptor. What happens afterwards? Cell becomes warned. Can we use interference to treat infections? We can. Is it safe? Not really. It's pretty terrible. So adaptive. Do you understand the idea of systemic response? Yeah, I can give an example so it sticks better. When you have a cut, okay, cut, the edges of the cut, how do they look sometimes? Color-wise? Red. Basically swollen. Okay? When you have a flu, how do you feel? Huh? Like shit, right? I've heard like shit, like crap, like you've been hit by a train. Yeah. That is because your immune system actually tries to destroy the pathogen. Okay? So in first case, you're fine. There's a little redness here, right? That's local. That's innate immune response. In second case, your entire body hurts. That's systemic response. Make sense? See the difference? Immunological memory. Self-explanatory. Before we get exposed to the pathogen, do we get any immunological memory? Like intrinsic? No. We're naive. We need to get exposed. Humoral cell, or B cells, T cells. T and B cells maturation. Know where they mature. They both originate in bone marrow. B cells mature in the bone marrow. T cells in the thymus. They both go into the lymphatic system, and then from lymphatic system, after the exposure, they enter the circulation. Antigen. We discussed the antigen. Are we clear about the antigen? Antigen, antigenic determinant. Is it on the antigen or on the antibody? I'm not asking any trick questions here. Antigen. Antigenic determinant is a part of antigen. We good? You see the difference between hapten and complete antigen? Hapten is small. If you stick it to something big, it can become antigenic. B cell, it's not positive. It's, okay, fuck this. Should be negative. B cells, what they must not recognize. Again, what they must not recognize. Cell, right? Know the process of selection, expansion, what they differentiate into plasma cells and memory cells. It's basically. Now, for receptors, know the structure of antibody. I told you before, I'm telling you again. Know the basic mechanisms the basic mechanism for VDJ recombination. Okay. I don't know what to tell you more here. 5 types of antibodies. Know them by heart. Okay. I will ask you the function. Okay. 5 major antibody types. Primary, secondary B cell response. Immunological memory, how it works. When you expose to the antigen second time, memory cells will mediate the response the most. They will produce plasma cells much faster, the higher levels. They will produce much higher levels of antibodies, and the humoral response will develop to be more robust and quick. Antibody profiles. Primary response. Remember, you have IgM, IgM first and IgG second, right? That's the primary. What happens in the secondary response? You're going to have overwhelming amount of what type? Huh? IgG. In the secondary, it's going to be overwhelmingly IgG. Humoral immunity. Active, passive, natural, artificial. Cool. Mechanisms of antibodies, how they work. Again, I want to remind you about opsonization. Okay. Neutralization, agglutination, precipitation. Are we familiar and comfortable with these terms? Do you understand what happens during each? Can antibodies kill the cells? No, they can't. Then it's basically, can antibodies activate the complement? Can antibodies that are bound to the pathogen, can antibodies activate the complement? Yes, they can. Okay. MHC. So we mentioned this. You need antigen to be presented to T cells. Does that make sense? 3 types of antigen presenting cells are dendritic cells, B cells, and macrophages. Which MHC molecule will present antigen to CD8 cells? No, CD8. Remember the rule, it's always 8. CD8 will be presented by MHC class 1, and CD4 will be presented antigen by MHC class 2. Okay, we're good? Repetitive generation, maturation, activation. Which organ? Juices in bone marrow, thymus, lymphatic system, blood. Can T cells be activated by the antigen itself? No. How does the antigen presentation happen? By antigen presenting cells. Positive, we didn't talk about positive selection. Don't worry about positive selection. Again, negative selection sends the T cells that can recognize our own tissues into apoptosis. Does it make sense to you? Good. So obviously this goes away. What if negative selection process fails? What if thymus fails to eliminate T cells that can recognize our own tissues as before? What are we going to get? Autoimmune disease. Functional types of T cells. The ones you should be absolutely familiar with. T helper cells and what they do. what they do, they help B cells to produce antibodies. They activate macrophages. They help to activate cytotoxic lymphocytes. T regulatory cells that control the immune response, and cytotoxic lymphocytes that can kill infected cells by inducing apoptosis, either via Fas ligand Fas receptor interaction or by releasing perforins and granzymes. Are we clear? What is the MHC restriction? Who remembers? Which type of MHC presents the antigen to CD4? 2. To CD8? 1. Can it be the other way around? No. That's MHC restriction. Does it make sense? CD4 cells are presented by MHC2. CD8 cells presented by MHC1. That's the definition of MHC restriction. Connection between helper and killer cells is helper cells are required for proper activation of killer cells. T regulatory cells control inflammation. What if you have no Tregs? So immune response goes on and on and on and on. What kind of disease are you going to develop? Autoimmune. How T cells link cellular and humoral immunity. T cells help to activate B cells, right? T helpers. And T cells, when they help to activate macrophages, that's the connection between adaptive and innate. Can cytotoxic lymphocytes phagocytose? Can they? Can cytotoxic lymphocytes phagocytose? There's only one answer. No. What are the two phagocytic cells then? Neutrophils and macrophages. So if T cells cannot phagocytose, they don't have a capacity for it. Advantages, I'm not going to ask you about advantages, disadvantages. We had a wonderful conversation, but no. Okay. 4 types: subunit, vector, inactivated, attenuated. We clear? Any questions about it? Moving on. 4 types of hypersensitivities. I got you these ones. Now let's, following clinical, so hay fever and anaphylactic shock. What type of whatever? Yeah, hypersensitivity, 1. Transfusion reaction, rhesus conflict. Lupus, TB test, poison ivy. I always tell you, every study guide is the closed list, which means I will not ask you what are the mechanisms underlying multiple sclerosis or rheumatoid arthritis. We mentioned it, but it's not there. Good? Moving on. Again, blood groups now know this. How you can prevent the hemolytic disease of newborns. How ROGAM antibodies work. That they basically mask baby red blood cells from mother's immune system. Transplant. We got this. We got 39. It's perfect. We covered it. Talked about it multiple times. Immunodeficiencies. I will be very clear, and I'm not going to throw any curveballs at you with immunodeficiencies, I promise. So in the list of SCID, cancer, flu, and A-gamma globulinemia, which 2 are acquired? Which 2 can be considered acquired immunodeficiencies? SCID, cancer, flu, A-gamma globulinemia. Cancer, obviously, influenza, depresses your immune system. Okay? Does that make sense? I will be very, very, very clear. Okay. Lymphatic system. Know where lymph originates. It's blood. When plasma leaks into the tissue, goes through the tissue, from the tissue into the capillaries, collecting vessels, trunks and ducts. Okay, goes through the lymphatic vessels. What controls its entrance into the lymphatic system? Remember we talked about mini valves at the capillaries? That's that. Lymph node. Features to focus on. Number one, you have more afferent vessels, incoming vessels, than efferent vessels. Does that make sense here? So that allows lymph to stay stagnant for a little bit. Does that make sense? Okay. So since lymph is more stagnant into the lymph nodes, since the flow of lymph through the lymph node is slower, better immune responses. B cells, mostly in the cortex. T cells, mostly in the medulla, macrophages. Medullary sinusoids. Okay. Yeah, lymphatic vessels, just, those four types. Capillaries, collecting vessels, trunks, ducts. Similar to veins. Where ate this? What a talented man. I'm talking about the diffuse lymphatic tissue. Okay, so let me appropriate of whole organs. Sorry about it. So for spleen, know the function, gets rid of red blood cells and white blood cells and platelets, and can release them too. Okay. Function of thymus, maturation of lymphocytes. Know specifically about the thymus blood barrier. Understand why it is important. Remember, thymus has no B cells, okay, and T cells are isolated from the blood to ensure that they do not develop tolerance to any pathogens. Okay. Mucosa-associated lymphoid tissue, where can you find it? In the mucosa of whole organs, like respiratory tract or like tonsils, okay, or the digestive tract, sites of the potential entry for the pathogens. Okay? I think we went pretty thorough